This presentation reviews the findings of Dr. Michael Bowers in his study, FOXP2 mediates sex differences in ultrasonic vocalization by rat pups and directs order of maternal retrieval, conducted in 2013. Communication is arguably one of the most important characteristics of an organism, allowing organisms to be competitive and survive. A more complex form of communication tends to align with a higher evolved organism. Humans, for example, have an extremely complex form of communication that involves many different languages and ways to indicate thoughts and feelings. We are able to communicate when we are hungry or thirsty, when we're scared, or when we are happy or sad. These reasons for communication are just as helpful to animals as they are to us, as they assist in survival. Animals can communicate with each other when there is food present or when they are in danger. Young offspring are able to vocalize their distress, allowing their parents to hear them and respond appropriately. Analysis of birdsong has led to developments in the understanding of the effect of sex differences on communication. However, there is not a lot of research on the implications of this discovery. Do sex differences in language exist just for mating purposes, or are there functional consequences of the ways males and females communicate? Is a mother able to understand the differences between the cries of her male and female pups, and if so, does she respond differently? In order to study language differences in the brain, FOXP2 must be used for analysis. FOXP2 is a protein that is a transcriptional factor of DNA and allows for the regulated repression and expression of genes. The expression of FOXP2 can cause alterations in neuritic outgrowth, dendritic branching, and the morphology of axons. FOXP2 is crucial for language development in an animal, and mutations in the gene can cause serious language deficits in humans, including but not limited to stuttering, autism, and SPCH1, or speech-language disorder 1. Interestingly enough, there's a great expression of FOXP2 in females, which could indicate why females tend to have stronger language skills than males. The prevalence of autism, for example, is much greater in males than females. In other animals, FOXP2 is just as critical for language. Zebrafinch songbirds have been shown to have increased FOXP2 levels during song development. When there is a reduction of the gene, songbirds have more variable song and are unable to imitate other birds. In this study, there were two specific goals. First, the researchers wanted to identify if there were sex differences in distress calls and determine what mediates this difference. Secondly, once this information was obtained, the researchers wanted to determine if the mother was able to identify these sex differences in calls from her young and if she responded differently to different sexes. The first goal of the research was to explore the sex-based differences in language. In order to examine the language differences, the amount of FOXP2 needed to be analyzed in male and female rat brains. Rat pups were bred and then killed at 4 days old. The brains were extracted, and Western blot analyses were performed using standard procedures on the cerebellum, amygdala, cortex, thalamus, hypothalamus, hippocampus, and preoptic area in order to detect FOXP2. The number of neurons in these areas was also counted. The examination of the FOXP2 in the brain allowed for a molecular view of how language differs between sexes, but in order to see if these differences were physically visible, another test was conducted. Rat pups 4 days old in age were removed from their mother, placed in a plexiglass container, and were allowed 5 minutes to make noise. Their calls were recorded and quantified using a threshold-based algorithm to detect ultrasonic vocalizations. The calls were analyzed based on frequency, mean amplitude, frequency bandwidth, duration, and number. Language differences were also analyzed based on how the sex of the rat responded to siRNAs that would damage the FOXP2 gene. The rats were injected with 1 microliter of FOXP2 siRNA at 1 mm caudal to bregma and 1 mm lateral to midline on both hemispheres. In order to view the effect of the siRNA on the FOXP2 gene and language expression, Western blot analysis and vocalization recordings were collected again using the same procedures, this time using rats with the siRNA implantation. The second goal of the research was to see how these language differences would be perceived by the mother. In order to do this, the pups were marked up for sex upon birth, and then the litter was removed from the nest after they were 4 days old. The pups would vocalize their distress, and the mother would come and carry them back to the nest. The pups were ranked based on the order they were brought back in, making note of what sex was carried back faster if there was one. This data would indicate if the mother was able to tell the difference in vocalization and was selective based on those vocalizations. While rats were the subject of the majority of the research done, human tissue was also collected and analyzed to view FOXP2 differences in girls and boys. In order to determine this, tissue samples were obtained from the brains of young children. PCR was used to replicate the FOXP2 gene in the human brains. Cortex samples were analyzed using the immunohistochemistry, and the amount of FOXP2 was quantified. There was no siRNA analysis of the human brains. Based on the analysis of the brain slices of rats, it is clear that the male pups have higher levels of the FOXP2 protein. In the cerebellum, amygdala, cortex, thalamus, hypothalamus, and POA, the male rats had significantly more FOXP2. The other areas of the brain that were analyzed did not have applications to communication and therefore were insignificant. The number of neurons expressing FOXP2 was counted, so it could be determined if this had a role in the expression of the protein. It was found that there was no cell count difference between the sexes. Based on the data collected, it was concluded that the heightened amount of FOXP2 in males versus females is general and is not based on cells. The pup vocalization data was analyzed and quantified, demonstrating that the male pups vocalized almost twice as much as the females. Male pups also produced slightly lower frequencies and vocalized at a lower decibel than the female pups. Because the male pups had more FOXP2, this data makes sense and is consistent with the previous findings. Once the siRNA was introduced, there was a shift in the differences between male and female vocalization. The addition of the siRNA caused the males to vocalize significantly less, going from an average of 603 calls down to 466 vocalizations. Interestingly enough, the siRNA had the completely opposite effect on the females, who went from 350 calls to 465 vocalizations. The males produced higher frequency vocalizations with the addition of the siRNA, while again, females had the opposite effect and produced lower frequency vocalizations compared to their control. The same effect carried through to the amplitude of the calls, and males vocalized at a higher amplitude than their control with the addition of siRNA, while the females vocalized at a lower amplitude than their control. The siRNA was tested on cultured cells in order to determine its effect on the FOXP2, and it was found that the siRNA reduced the FOXP2 gene by about 65%. The siRNA had no effect on FOXP1. siRNA's effect on FOXP2 was evaluated in the cortex and the striatum, and was found to be effective in both areas. When examining the siRNA's effects on the FOXP2 in males versus females, it was found that the FOXP2 expression greatly decreased in the males. However, the expression increased in females, explaining the vocalization findings. This suggests that a rebound in the expression of FOXP2 at PN4 followed the inhibition at PN2, not seen in males. Based on the data obtained in the previous experiments, the scientists hypothesized that the mother would retrieve the male pups first because of their increased vocalizations. The hypothesis was supported by the data, and the males untreated with the siRNA were recovered before females untreated with siRNA. However, once the males and females were treated with the siRNA, the data resembled that of previous siRNA data, and the females were recovered first. When evaluating the human brain tissue samples, scientists discovered that females had a high basal level of FOXP2 compared to males. There was no significant difference in the levels of FOXP1. Based on the data collected, it can be supported that there is an innate sex difference in FOXP2 levels, causing differences in communication between the sexes. There may be a variety of reasons for this that could include either genetics or hormonal exposure, which could be explored in future studies. When comparing the FOXP2 in the brains of male and female rats, it was clear that the males had a greater amount of FOXP2, and subsequently were able to vocalize more efficiently than the females. However, upon siRNA injection, the males had reduced FOXP2, and the females had greater FOXP2. Considering that the siRNA was supposed to reduce the FOXP2 in the brain, this finding is a bit surprising. The researchers suggested that these results could possibly be attributed to an overshoot caused by the low baseline of FOXP2 in the female brain, though more research will be necessary to further explore these conclusions. The second goal of the research was to examine if the mother was able to identify the sex-based language differences with their pups. The results confirmed that the mother rescues the pups that have increased vocalization. She may do this to balance the sex ratio based on the environment. Essentially, all the rodent studies demonstrated that the male rats had an increased level of FOXP2 and an increased level of vocalization, indicating a more advanced communication level. However, when the human brains were tested for FOXP2, the opposite was found. Females had a much higher level of FOXP2 and are known to be better communicators than boys. The reasons behind these differences between the rodents and humans are not known. The experimenters did not note any limitations that they experienced during their research. However, they did explore areas for future research. One of the most intriguing findings from the study was the discrepancy between rat female brains and human female brains. There is no known explanation for why human females have significantly more FOXP2 than rat females. More research needs to be conducted on other mammals to see the results for the female brain and to help explain why this phenomenon is occurring. Furthermore, in this study, FOXP1 was not found to be significant for the communication of rats. However, in humans, it is known to be closely related to language abilities. It is possible that the addition of the effect of FOXP1 may have an impact on the human brain communication skills, and this area should be examined more through research. In this study, the researchers focused on a young age for both the rats and the humans. If they had examined different ages that were correlated with certain stages of development, they may encounter some different findings that could be useful. Furthermore, the researchers could have used more samples. While the availability of human brain tissue is not high, the sample sizes that they used were very low and only included 5 males and 5 females.